Right, I have the great honor to introduce the first keynote speaker of the conference, Dr. Pamela Lyon. Pamela is an independent scholar. She's affiliated with the University of Adelaide, following a decade's association with the Southgate Institute for Health, Society and Equity at Flinders University of South Australia, which no longer exists. Pamela received her undergraduate degree from the University of California, Berkeley, a master's degree in journalism from Columbia University, and her doctorate at the Australian National University, where her radically discipline-crossing thesis won the university medal. Pamela is a natural philosopher. She develops theory and conceptual frameworks in the biology and evolution of cognition, from prokaryotes to humans, and she's particularly interested in the coevolution of cognition and stress responsive. Her approach is to use scientific research to answer questions that have poorly conceived answers or plausible answers that are only heeded by few. What is cognition? What biological function does it serve? How did it evolve? And what relationship does it have to life? One of the things that inspires me and interests me the most, and I appreciate the most about Pamela's work, is her approach and insistence on focusing on the simplest model systems possible first, and only later extending it to more complex ones. All right, so without further ado, please join me in welcoming Pamela Lyon to the online stage for her talk.
Thank you.
And
thank you so much for that wonderful introduction, Eduardo. Thank you very much. And greetings to everyone from Jerusalem, which I had hoped to be there, but unfortunately, my travel did me in. My ghost was not able to keep up with my body. So the talk I'm going to give today is called Who's Afraid of the Ghost and the Machine? Especially if the machine can't run otherwise. The artwork I have here is by a collaboration of Genetic Moo and the Creative Collective in the UK, which is called the Tangled Bank, and is a reference to Charles Darwin's poetic ending to The Origin of Species. And what I'm going to hope to bring home to you again and again through this talk is that it's looking more and more as though life really can't function the way we know it does without cognition. And through an adaptive plasticity that is unusual in anything we know. It's very, very, very odd, weird in some respects. And so what I'd like to say, I'd like to give you the take-home messages first so that you can follow along, you can try to keep them in mind as we move through the material. Now, the first one is that life really isn't possible without the processes traditionally considered cognitive. And I will take some time to go through a series of premises and conclusions that I drew out of the literature that led me to this conclusion. Secondly, ALIFE definitely should take account of the cognitive in biology. And I think that in actual fact, it is going to help ALIFE considerably because you will be able to enlist parameters with purpose rather than hoping that the simulations are going to somehow deliver it for you, when you'll be able to bake it in your work. That's what I'm hoping, at any rate. So how on earth did we get here? How did we get to talking about something called the ghost in the machine? Well, the person who coined this phrase was an Oxford philosopher who was extraordinarily influential and was the editor of Mind for years, named Gilbert Ryle. But he basically blames someone else. He says that the person we really should be sheeting all this home to is René Descartes. And that is because Descartes gave us what Ryle called the official dogma of the ghost in the machine, and that is mind-body dualism, which is the idea that mind stuff and body stuff are two different stuffs, and his view is that this may seem like a logical way to think of things in a common sense way, but he is adamant that it's false. And it's false. He bases his attack on 3 lines. The first one is philosophical, arguing that the metaphysics are confused. I think it's a very bad argument, so I'm not going to rehearse it here. The central one is linguistic, saying that mind as a concept is a category mistake, in the same way that if you had gone to universities and someone showed you all the buildings, all the staff, all the students, all the sporting grounds, and then you said, oh, that's all well and good, but where's the university? And he said, that's basically what we do when we look at behavior and that sort of thing, and then say, where's the mind? Because in Ryle's view, he was a complete behaviorist. Behaviorism was ruling the day in at least North American philosophy, in North American psychology. And so he was saying the behaviorists don't need a concept of mind, and except for our folk interactions with one another, we don't need one either. And but he was so adamant about this that he really basically wanted to eradicate it completely from any kind of philosophical or scientific discourse. And the reason for this is vitalism, which was associated with superstition and religion, and above all, ghosts. But it also was reflecting a battle that was then really quite heated for the soul of biology between vitalists and mechanists. I have put these in scare quotes because these are terms that do not bear much scrutiny according to the way we now think of these things. But basically, from the comparison of their views was these became ideological forces in biology that really just got completely out of control, as far as I can see. But the vitalist view was that life is different, that unlike the mechanists claim that life is physics and chemistry, and it will yield to explanation in the same way that physics and chemistry have. The vitalist said, no, not really. Self-organizing, developing system need new kinds of explanations. Whereas the mechanists were saying reductionism work, physics and chemistry work, no matter how complex these systems are, they will yield to these kinds of explanation. And the vitalists were also saying that look, there's multiple kinds of vitalism. There's a methodological vitalism, there's epistemological vitalism, and they don't necessarily equate to metaphysical vitalism. Metaphysical vitalism is a belief. Whereas the mechanists were saying physical and chemical processes are the only real furniture of the world, and they believed that science so completely endorsed this view that they could cease to say that it was a belief, that it was actually true. So, but what Ryle forgot about Descartes was that Cartesian dualism carried with it a machine view of life that held not only that humans are the only ones that have mental lives, the only ones, that the rest of the living world is mindless and machine-like. So subtracting mind leaves only the machine, which raised no end of problems for naturalizing the mind in the latter part of the 20th century, when people could talk about the mind again, when behaviorism had basically been put in its proper domain and not as the sort of imperialist that it had become. And it also raises problems for explaining cognition. And the person who was chained to this legacy for his very long career was Daniel Dennett, who was absolutely bought into Ryle's crusade to drive a stake through the heart of vitalism, and that's because Ryle was his teacher. Okay, so meanwhile in Australia, I was a PhD candidate in the Faculty of Asian Studies at the Australian National University, trying to puzzle out a cross-cultural philosophical analysis of some propositions in Buddhist philosophy that were considered established. And what I wanted to do was use Western philosophy and Western science to see if that was in fact the case. But what I forgot was that almost nothing in Tibetan Buddhist philosophy is completely divorced from cognition, consciousness, awareness, and mind, which are all synonyms in the Buddhist tradition. And so I needed a comparator. And so I went and I looked at cognitive science. And judging from this list here, there was almost nothing here that I could relate to. The extremely detailed and rich tradition of mental functions and activities to the kinds of things I was seeing in cognitive science. When I looked more closely, however, it seemed to me that the common denominator of all these things, when I looked into them closely, was it a starting point in human cognition. And it was that starting point in human cognition which allowed people to just think, well, I've got a mind. I've got these intuitions about what my mind is like and what minds are like. And because my mind has got to be like other people's minds, then my intuitions about my mind should be valid. But they weren't necessarily. And I gave this a label because it started in the human case. I called it anthropogenic. And the big question I had was, where's biology and evolution in all of this? Now, embodied cognition was just really getting some wheels at that point. But it seemed to me that it was just an acknowledgement of the bleeding obvious that cognitive agents as we know them are embodied. And, it was really about time. And somebody started mentioning this, and it was hardly news. So I didn't take it that seriously as a program, actually. I thought that biology would get us there. The other thing that I really noticed about these anthropogenic approaches is that there were very few constraints on theorizing and many false positives, and they ended up in endless debates about where cognition lies in the world. But the one place you couldn't say that it existed was in anything simple. And because biology really didn't figure in most theorizing at that time. So what that left me from the Western educated, industrialized, rich and democratic world was a concept of mind or cognition that was undefined, that it was associated with an organ of the body, but and what it did were these things that I had never thought of before and didn't make any sense to me. Computation over representations, information processing, fine. Clusters of capacity, sensing, memory, learning, decision-making, yes. And implements movement and behavior, not a problem, but try finding, identifying criteria for identifying mind or cognition in the natural world. Effectively, there were none. So basically, I had no comparator. So I then looked at what biological approaches were suggesting, and I looked at a range of different frameworks, and I'm happy to talk about what those were in question time if you like. But what I found was that all of these things that dropped out of biological approaches were really hard constraints on theorizing. So I named this the biogenic approach with some relief and noticed that embodied cognition was included in this already. Embodiment was assumed and situatedness was assumed. The issue of extended mind, that is something that would have to be determined on a case-by-case basis. But I didn't see that was necessarily intrinsic to cognition as a biological function. It might have arisen out of that, but it wasn't intrinsic to it. Here are some biogenic principles. This is just, I'm showing this strictly as an illustration of the ways in which theory really was constrained by these principles in biology. And what these yielded for me were three basic facts of life that I then took to be the implicit existential goals of all organisms. The first of which is that biological systems, organisms, seek to persist or survive. They are dynamically self-preserving. Second, organisms engage in action to facilitate their growth or well-being. No surprises here. Thirdly, organisms engage in action to facilitate their reproduction. This is all really standard stuff, but no known non-organic, self-organizing, autocatalytic system can do these things. They can persist for a period until they run out of fuel, but then they stop. They could reproduce themselves, but it is not an impulse of the system. And growth, who cares? So out of these frameworks, I came to think of these things as premises in an argument that had conclusions. And I'm really sorry if I'm boring you with this too much here, but it's really important that you see how cognition basically falls out of the lived state. So premise one is that organisms maintain themselves far from thermodynamic equilibrium by importing matter and energy from their surroundings, chemically transforming them to produce highly organized structures and processes that sustain the system, and exporting disorder in the form of waste products. Premise 2 is that organisms must establish causal relations with features of their surroundings that lead to exchanges of matter and energy, which are essential to the organism's persistence in the first place. Premise 3, organisms are continually being produced, not just maintained, not just self-organizing, produced by a network of components which are themselves being produced by networks of components. At the same time, the organism as a whole, as well as its constituent components, are interacting with a surrounding medium. Premise 4, organisms are constituted by a wide variety of regulatory mechanisms, including multiple kinds of feedback, which maintain system parameters within a range that will ensure system persistence, homeostasis or allostasis. Premise 5, to persist, grow, thrive, or reproduce as conditions change, an organism must continually alter its internal structure and or its interactive relation to features of the surrounding medium and changes in internal processes, constantly altering the relation between the outer and the inner, which is something that Herbert Spencer noted in his Principles of Psychology in the mid-19th century. Premise 6, a state of affairs that stimulates an organism to adaptive behavior, that is a change in its internal structure and or its interactive relation to its surrounding, conveys information for that organism. It is, in Bateson's terms, a difference that makes a difference. Conclusion 1, because they're thermodynamically open autopoietic systems, every organism requires a means of determining the interactive potential of its surroundings. It needs to be able to monitor externally. Because every organism requires a means of, it does these things, it also requires a means of determining its own status, its level of functioning relative to continued autopoiesis. Conclusion 3, to persist, all organisms therefore require a means of relating information about internal states to information about external states. This involves valuing what's happening now as acceptable, advantageous, or dangerous. It also, that valuation is, because it is always a projection of value relative to the state of the organism, it has a perspective. It has a kind of intrinsic subjectivity. And because they're not assessments of objective states of affairs, these projections of value are subject to error. Conclusion 6, because such valuations are subject to error, every system also must be capable of ascertaining the failure of an adaptation to try something else. 7, and now we're getting close to it, staying alive requires a suite of capacities considered cognitive in humans, including sensing, that's internal and external monitoring, valence, is this acceptable, advantageous, or harmful? Memory, what happened just a moment ago compared to what's happening right now? Discrimination, that this is this and that is that. Some form of learning and the ability to integrate multiple sources of sensory information to make decisions about what to do next. Finally, because a living system is endogenously active and constantly in need of new sources of matter and energy, it is temporally future-oriented. What comes next? Systems contributing to organism fitness at many levels are therefore likely to be anticipatory. Okay, so my question was, where do you find it? So I went straight to the bottom and looked at bacteria and expected, in all honesty, to rule them out. But what I found at this level was so gobsmacking, was so amazing and so interesting. I am still there 20 some years later. No, almost 20 years later. I thought I'd be onto vertebrates by now, but I'm not. And the reason why is bacteria seem to be the gift that keeps on giving. And this is the cognitive toolkit that I have now worked out based on my explorations in the micro world. All of these capacities are found in bacteria. If you have a quick look at my definitions, and there is only time for a quick look, you will see that they're very simple. They do apply to human beings, but they do not depend on the human case. This is sort of like the minimal version of what these capacities are. Okay, this approach led through a workshop in Vienna in 2018 to the emergence, the beginnings of a field that is now called basal cognition, the aim of which is to see whether, per Darwin, whether an evolutionary continuum of cognitive function exists from the very simplest to the most complex of living things, to what extent and how it's implemented. That's the goal. Basal refers to the models of study, usually on the lower or evolutionarily earlier branches of the phylogenetic bush. In general, basal refers to unicellular plants and animals without nervous systems, or aneural organisms, animals with very simple nervous systems, such as hydra, worms, and larval stages of various organisms, and intraorganism systems like cells, tissues, the immune system, development, and regeneration. Again, in my way of thinking, basal cognition is not a form of cognition. It is a reference to the models employed to explore cognition. Now, basal cognition's most prolific theorist is Michael Levin, who's many of you may be familiar with his work, so I will only briefly mention it here. I suggest you take a look at the presentations on his website. As many of you know, Michael is Mr. Bioelectricity. His first claim to fame was torturing, I don't know, treating planaria with electrical charges that then induced two heads on a regenerating planaria, which was then maintained subsequently in subsequent regenerations without any changes in the genome. This was entirely adaptive plasticity. His work with the African clawed frog has been similar in this notably. He and his crew altered a tadpole of a frog in ways that really distorted its developmental program, but then the developmental program produced a normal frog, which again is adaptive plasticity. And from those frogs, he and his colleagues, including notably Josh Bongard, developed something that they call xenobots, which are like living robots that seem to have their own impetus. They're programmable to do certain things, but they also their behavior is extraordinarily cognitive seeming. I would say cognitive, but not everybody would agree with that. But that's Mike's work, and I work with unicellular, so we're going to have to go back to them. Now, we're going to start somewhere fairly simple. It's not simple at all, actually, but it is the ground floor of any kind of cognition, and that is even before neurons, protein-based computing with chemistry. And what we now know is that this computational mode is extraordinarily powerful. According to Sterling and Lachlan in their Principles of Neural Design, simple chemical circuits are capable of implementing procedures used in analog electronics, like amplifying, oscillating, differentiating, and integrating, as well as taking laws and raising to powers. Chemical circuits support arithmetic operations as well: add, subtract, multiply, and divide. And without being more specific, they say small chemical circuits also have the ability to perform more complicated functions. Now, this is a wide repertoire that cells have. What we didn't expect was that cells, free living cells, would have these organs at their leading poles that are large protein arrays of chemoreceptors that can sample existentially salient chemicals from the environment. Each of these organs has several thousand chemoreceptor proteins, and they are generally at the leading pole of the cell, which is why Jeffrey Stock and his colleagues at Princeton called them nanobrains. They lead the cell, and they are signal processing organs that are, this is dedicated hardware, and therefore is a bit more costly than your normal signal transduction. So there is some issue about whether something else might be going on. And when you look at what's inside these things, it's a serious question, because each of these chemoreceptors are trimers of dimers. And when you look at the dimers, that's the two arrows, they have 8 methylation sites, each dimer. And there are 3 of them in each side of these chemoreceptors. So think about it, 10,000 chemoreceptors, each with 24 to 48 different methylation sites for registering different situations, conditions in the environment. Now, people have pointed to this and said, well, all they do is basically turn flagella, so they can't be all that important. But I suspect because they are so energetically costly that there is something going on here that we really don't know yet, and we'll see. Okay, but bacteria now apparently have electrical signaling as another mode. Now, we've known since the 1980s that bacteria have ion channels embedded in their membranes. Roderick McKinnon used an ion channel in Streptomyces lividans to elucidate the structure of the and mechanism of the ion channel, which led to the 2003 Nobel Prize for Physiology and Medicine, because it sketched out in empirical terms Hodgkin and Huxley's proposed model. But at the time, no one had the faintest idea what ion channels do in bacteria. That is until 2015, or the year or two before 2015, when a research group spread across the University of California at San Diego and Barcelona, headed by these two remarkable men, discovered that ion channels in Bacillus subtilis have the same function as neurons, and that's to signal rapidly over long distances to large numbers of cells. And just to a fun fact, the leading model for investigating the mechanism of anesthesia right this minute is a bacterial voltage-gated sodium channel only found to exist in 2008. Now, does this particular sodium voltage-gated sodium channel immobilize the bacterium under the influence of anesthesia? We shall see. Okay, here is an example of how electrical signaling has been shown to work in Bacillus subtilis biofilms. A biofilm is a structured community that has two principal zones. The first one is the center, and the other one is the periphery. And now the center can get very congested and very constrained, but at the peripheries, the cells are munching away at what nutrients are available and reproducing and expanding the colony. Now, at some point, the cells in the center become highly stressed. And when they do, they trigger a metabolic positive feedback loop that induces a release of potassium through action potentials. This depolarizing wave stops the cells on the edges from feeding, which allows nutrients to diffuse into the center. So they basically take turns eating. Now, this would be unusual enough in itself, but we now know that this is how biofilms that are spatially separated from one another also share diminishing resources by taking turns feeding. It's like time sharing on diminishing resources. And that is also through electrical signaling. And what's more, electrical potentials have also now been found to be involved in the germination of B. subtilis spores. Now, the mechanism by which bacterial spores germinate has been a mystery for a very long time. And the fact that it involves electrical potentials is pretty strange. Okay, there is also interesting predictive regulatory networks in E. coli and also in yeast. E. coli is a bacterium that has two general habitats, one environmental in water, soil, and and the other one in generally a mammalian host. So starting here, E. coli infects a crustacean that is then consumed in a prawn cocktail by a human being, which starts a cascade of a sequence of two stress responses. When the bacteria enter the mouth, there is a temperature rise that is natural because mammals are warm-blooded and their temperature is maintained at a higher level. And so the bacterium triggers heat shock proteins and other heat shock mediators. And shortly after that, that is a signal for another stress response that is to deal with the drop in oxygen level, which is predictable when it goes into the digestive tract of the host. So the bacterium switches from aerobic to anaerobic respiration. Now, the thing that is amazing about this is that they are dissociable. And when Tagopoulos and colleagues at UC Davis conditioned these bacteria out of phase, they were able to reverse the temporal sequence over generations, which is suggestive of epigenetic inheritance. That means they changed the order of them, so the oxygen level dropping became the prediction for temperature rising. So they were dissociable. Now, these kinds of anticipatory regulatory networks are being found a lot now. Not everybody is willing to go the extra mile to dissociate them and reverse them, but this is not something that is a one-off in E. coli. Now, the basics of learning have been known in bacteria for a very long time. That's habituation and sensitization. Apparently, when habituation was demonstrated in E. coli, neuroscientists basically had apoplexy and said this could not be, but it can be, and it is. But whether or not they're capable of associative learning, nobody has yet tried to attempt that, but it has been attempted in amoeba and with astonishing results. This was based on a pilot study. There was a large scale series of experiments involving 9 academic entities in 5 labs in 3 countries, Spain, Israel, and Russia. And there were 4 reviewers on this article that was published in Frontiers. The experimental treatments included a control of normal feeding, an electric field to determine what the natural orientation were of these 3 species of free-living amoeba. This included, and they were found to be naturally attracted to the cathode. So they were also tested on their chemotaxis toward a molecular signal for prey bacteria, a peptide. And the induction was a simultaneous presentation of an electric field and the chemotactic peptide at the anode. And then the conditioning test was an electric field without the peptide. And their responses to the conditioning test in more than 2,000 free living cells that were tracked individually, they measured response intensity, directionality and persistence times, distance traveled, directional ratio, and average speed. They did this by measuring them and washing them and then testing them again and then washing them and then testing them again and using a lot of cameras. What they found, was that here you have the unconditioned cells that are all migrating toward the cathode very, very dramatically. And here are the conditioned cells without the chemotactic peptide migrating considerably to the anode. Now, we don't know what galvanotaxis does in the ecology of amoeba, but this is an extraordinary result. Okay, so how is the battle for the soul of biology going now? Well, we go under different names, but things have changed quite a bit from the time of Ryle and early cognitive science. Or they're called the people who would have been called vitalists in another time are now rightly called organicists, and they have been fully vindicated in their assertion that life is different and it does require new kinds of explanations. And it is also really clear that you don't have to be a metaphysical vitalist to think that life is different and is going to produce new kinds of explanations, and that physics and chemistry aren't going to get us there. Now, the mechanists are now called physicalists because the organicists believe in mechanisms, but they still assert that life will be reducible to physics and chemistry. But this is now clearly just a belief. As for reductionism, that complexity will yield to physics and chemistry, the methods of physics and chemistry, only with new concepts and tools. And physical chemical processes are the only real furniture of the world. This is now clearly a belief. There is no lay down evidence for anything really at this point. So we started out with a quote from the account of Gilbert Ryle and what the ghost in the machine meant. But there was, as has been mentioned, another man, an East European philosopher and polymath named Arthur Koestler, who wrote an entire book that was a riposte to Ryle and the program of life is machine-like that grew out of it. And I want to leave you with this quote that is really not all that perspicuous, and I will unpack it in a moment, but I think it's important. A true science of life must let infinity in and never lose sight of it. The great innovators in the history of science have always been aware of the transparency of phenomena, not just life, towards a different order of reality, of the ubiquitous presence of the ghost in the machine. Now, what on earth does that mean? Well, first and foremost, it means that life tends toward complexity and integration in ways that things that are not living cannot. And they exhibit an adaptive plasticity whose limit, frankly, we do not know yet. When you have genome shattering under conditions of extreme stress in a process called chromothripsis, which then rearranges the genome in the hope that something is going to help, that is pretty weird. That's not just whole genome duplications, it's a whole different level of response. Koestler's main point is that organisms are not dualities of body and mind, but hierarchies of interdependent subsystems, which are both parts and wholes with some individuality, as he calls it, a kind of directedness in their processes. Now, these two things together are what yield the ghost. And that it is these two things together that makes him say that the ghost in the machine is ubiquitous in nature. I would say that cognitive processes are ubiquitous in nature. Now, physicists of the 20th century had a lot of difficulty coming to terms with the quantum world, but they now routinely work with it without thinking twice. At the molecular level, life has always worked with the quantum domain. It has always been transparent to a different order of reality, even if we didn't see it. So the most productive tool for anyone in attempting what you are attempting is an open mind. Now, I leave you with dish brain. The human and mouse brain cells that have learned how to play the 50-year-old video game Pong in a Dish. Now, the first article that announced this feat was in Neuron in October of last year, but has only recently this month reached into the mainstream media because of a large amount of money provided by some investors to power this up. And including $600,000 from the Australian government, because this is an Aussie startup. Now, I'm going to leave you with, just for this moment, a look at the dish brain system and experimental protocol schematic. I can't explain it to you because I've only just discovered it and I've only just skimmed the article. I really don't understand it well enough yet. I think that probably many of you, just by looking at this, will probably understand it better than I do, or I could right now. Now, you know, I'm perfectly happy to send this to you, but it's there in the article. And with this is my parting gift to you, because I think it is so it is such a good exemplar of the attempt to synthesize the organic and the synthetic in ALIFE. Okay, so I now need to thank the ghosts in my machine, the people who without which I could not function. And first and foremost is my dear husband of 36 years, without whose financial and emotional support this work would not be possible, but also this crew of individuals who have been really, really, really indispensable to the my development as an independent scholar. So thank you very much for inviting me. I'm sorry I don't know anything about computation, so I can't really answer any questions in that vein. But if you have any questions now, I'd be happy to field anything you've got. Thank you very much.